Good day, this is the Professor, CBMD PhD, and today's Saturday, May 27, 2023. It is 8:14 and 11 seconds, p.m. Eastern Daylight Time, and today's research, liver function. So let's jump to it here. Has cancer in distant organs alter liver function? Today's May 25th, 2023. Source: Weill Cornell Medicine. As cancer often releases molecules into the bloodstream that pathologically alter the liver, shifting it to an inflammatory state, causing fat buildup and impairing its normal detoxifying functions. And this discovery illuminates one of cancer's more insidious survival mechanisms and suggests the possibility of new tests and drugs for detecting and reversing this process. The researchers found that a wide variety of tumor types growing outside the liver remotely reprogrammed the liver to a state resembling fatty liver disease via secretion of extracellular vessels and particles containing fatty acids. As the scientific investigators found evidence of this process in animal models of cancer and in the livers of human cancer patients, the findings show that tumors can lead to significant systemic complications, including liver disease, but also suggests that these complications can be addressed with future treatments. As these effects reflect specific strategies cancers use to secure their survival and speed their progression, the team discovered that pancreatic cancers secrete molecules encapsulated in extracellular vessels that travel through the bloodstream, are taken up by the liver, and prepare the organ to support the outgrowth of new metastatic tumors. In the new study, the researchers uncovered a different set of liver changes caused by distant cancer cells, which they observed in animal models of bone, skin, and breast cancer that metastasize to other organs, but not to the liver. The study's key finding is that these tumors induce accumulation of fat molecules in liver cells, consequently reprogramming the liver in a way that resembles the obesity and alcohol-related condition known as fatty liver disease. The team also observed that reprogrammed livers have high levels of inflammation, marked by elevated level of tumor necrosis, factor alpha, and low levels of drug metabolizing enzymes called cytochrome P450, which break down potentially toxic molecules, including many drug molecules. The observed reduction in cytochrome P450 levels can explain why cancer patients often become less tolerant of chemotherapy and other drugs as their illness progresses. The researchers trace this liver reprogramming to extracellular vessels and particles that are released by the distant tumors and carry fatty acids, especially palmitic acid, when taken up by liver-resident immune cells called Kupffer cells. The fatty acid cargo triggers the production of tumor necrosis factor alpha, which consequently drives fatty liver formation. Although the researchers principally used animal models of cancers in the study, they observed similar changes in the livers of newly diagnosed pancreatic cancer patients who later developed non-liver metastasis. One of the more striking observations was that this extracellular vessels and particles-induced fatty liver condition did not co-occur with liver metastasis, suggesting that causing fatty liver and preparing the liver for metastasis are distinct strategies that cancers use to manipulate liver function. The scientific investigators suspect that the fatty liver condition benefits cancers in part by turning the liver into a lipid-based source of energy to fuel cancer growth. They see in liver cells not only an abnormal accumulation of fat, but also a shift away from the normal processing of lipids, so that the lipids that are being produced are more advantageous to the cancer. As that may not be the only benefit that cancers derive from this liver alteration, there are also crucial molecules involved in immune cell function, but their production is altered in these fatty livers, hinting that this condition may also weaken anti-tumor immunity. The researchers were able to mitigate these systemic effects of tumors on the livers by implementing strategies such as blocking tumor extracellular vessels and particles release, inhibiting the packaging of palmitic acid in the tumor extracellular vessels, M particles, suppressing tumor, necrosis factor alpha, activity, or eliminating Kupffer cells in the experimental animal models. The researchers are further investigating the potential of implementing these strategies in human patients to block these remote effects of tumors on the liver, and exploring the possibility of utilizing the detection of palmitic acid to tumor extracellular vessels and particles circulating in the blood as a potential warning sign of advanced cancer. And this is a professor of microbiology, bioinformatics, pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.